Hello, and we are back for part three of our look at some introductory information for the nervous system. And in this third video, we're going to be focused on looking at uh, some basics of an action potential. And we've already spent a lot of time on this, and so this is just going to be more of a review, just to make sure that you know how it applies and works within the nervous system. It is the same process. All right, it is the same process. And what I mean by that is things that you need to keep in mind, all right, that I am not going to explicitly go over because, again, we've already done this. And so I'm not going to kind of rehash it again. But kind of remember that the standard for resting membrane potential is negative 70 millivolts. All right, now there is variability in that. All right, keep that in mind. All right, there is variability in that, but negative 70 is the standard. That threshold is still viewed at being negative 55 millivolts. All right, we still have the all or none hypothesis. All right, that is still at play. All right, we still have depolarization that we need to contend with. All right, remember that goes up to between positive 30 and positive 50 millivolts. Right? We still have repolarization, right, which is characterized by an influx in potassium. Right? Remember, when we're dealing with depolarization, we're dealing with an influx of sodium ions. We still have hyperpolarization. And again, that is remember, an over influx of potassium ions. So all of these things, all of these standards are still in play. Right? Nothing here has changed. All right, and so we're just really looking at this from the standpoint of how does this apply to the nervous system. All right. So again, action potentials are going to be self-propagating once they have been initiated. So once that initial stimulus has triggered the initial action potential, that action potential will continue to self-propagate. Now, I want to remind you that you're really recreating that action potential. It's not the same action potential that has been strengthened. You are creating a new action potential at each influx of sodium channels that we run into. And remember, when we're looking at myelinated axons, that self-propagation is only happening at the node of Ranvier. That is it. Sodium channels are not present within the myelin, only within those myelin gaps or those nodes of Ranvier. All right, so that is something that you need to keep in mind. In non-myelinated axons, as long as you've got a sodium channel along that axon, it will go ahead and depolarize and repolarize. All right, and so you got to keep in mind that there is a difference. Non-myelinated works very much like skeletal muscle works. Right, myelinated, you've actually got gaps that are happening. Literally, it just can't reinitiate. There's some resetting that has to happen. So we need to look at that. All right, and the other thing is these action potentials only occur in one direction. right? And that's a forward direction. And we'll touch on why that is as well. Right? So we will get there. So let's look at a propagation of an action potential. All right, we can see here. Let me just change this over. Pointer. And so we can see here, right, we are at resting membrane potential. Right, we're at negative 70 millivolts, right, at the electrode. So here's the electrode. The electrode is measuring negative 70 millivolts. In other words, the membrane is polarized. All right. The action potential that's moving down along our axon, right, has not yet reached this electrode. Right, so resting membrane potential is good to go. Right, we have an area of action potential here. Right, we can assume that when we see these internal positive charges that we're somewhere between positive 30 and positive 50 millivolts. Right, in this view, hyperpolarization has not yet occurred. Right, so we go to the second image. Right, here's our electrode. Right, you can now see the action potential has reached the electrode. Right, what happened there? Let's assume that was a node of Ranvier. Right, what has happened here? Well, what has happened is positively charged sodium ions has moved into the membrane, which means just outside of that we have more negatively charged ions, right? Or I should say more negatively net charge over here. And it's like that on both sides. Right? You can see we've spiked up to about positive 30 millivolts up here. Right? Negative 55 isn't even on here. Right? It doesn't matter. And it doesn't matter because remember, all or none. It's either going to go or not. If you've hit negative 55, it's going to go. And that's all that matters. Right? That is all that matters. And so the fact that we've stopped the voltage, right, at positive 30 millivolts at 2 milliseconds tells us that we've hit threshold. Right? But look what's happening behind the action potential. Right? We've had hyper-repolarization occur all along here. Right? Here, we've got depolarization that's happened. Right? This is depolarization that's happening right here. Right? And ironically, what ends up happening here is these positive charges continue to displace these negative charges on the inside. Right? And that's the movement of the action potentials down the axon. So these positively charged ions here are going to displace all of this negative net charge, right, allowing that action potential to continue to move down this axon. And this is where we come over here. And again, at the area of the electrode, we have hyper-repolarization. Right? We've got polarization, right? Resting membrane potential here, hyper-repolarization here. Right? And you can see that our action potential is continuing to move down. Right? And that's really what was, that's what's happening. Now, this will continue to happen. Right? This action potential will continue to move down until it reaches a node of Ranvier, at which point in time the action potential will be repropagated or recreated. Right? So when we look at the intensity of a stimulus, right, or when we look at the speed at which that action potential is going to move, all right, what we need to keep in mind is that based on the all or none theory, it doesn't matter how strong the stimulus is, the action potential strength is always going to be the same. Right? As long as the stimulus is intense enough to meet threshold, everything else is going to move. Right? What will impact the action potentials is the frequency of the impulses. All right? It's the frequency of the impulses. All right? So how many impulses are reaching those sodium channels? And keep in mind that for the initiation of an action potential within an axon, we go back to the axon hillock. Right? That is where action potentials are generated within the neuron. Let me say that again. In fact, I'll do one better. I'll write that out just to make sure, because I don't want that point to be lost on anybody. It's an easy detail to skip. All right. Action potentials within an axon are initiated within the axon hillock. So if I draw my axon out, or if I draw my neuron out, so if I draw my axon out, right, or my neuron out, my dendrites are up here. Right, these are my dendrites. This is not a very pretty drawing at all. Right, but what I want to picture right here is there's the axon hillocks. Right, remember, it's part of the cell body, but it's the initial segment of the axon. And so it's kind of where the cell body and the axon merge. This is where the action potential is initiated, right there. Right, so those neurotransmitters that are being received by the dendrites come down and initiate action potentials within the axon hillock. Right, so don't let that be lost on you. That's where all of this is happening. So how many, the frequency of the stimulus, right, how many stimuli are received per second here is going to determine how fast those action potentials, or how frequently those action potentials are going to be sent. Right? Higher the frequency means a stronger stimulus. Right? So in other words, we're hitting boom, 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 boom. Right? High frequency. Right? You're going to have action potential, action potential, action potential, action potential, traveling all the way down here. And when it reaches that messed up axon terminal down there, what's going to happen? All of these neurotransmitters are going to be released. But if you have if the frequency is boom, 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 that's a slower frequency, which means those action potentials are not going to be as rapidly arriving down in the axon terminal. What does that mean? It means you're not going to be releasing as much neurotransmitter. So that is what we are referring to here. Now, it should also be noted that there is a reset period, right, what we refer to as a refractory period, that happens between each stimulus. Right? Again, this is no different than what we saw within skeletal muscle. Right? This is no different than what we saw within skeletal muscle. Right? Between depolarization and repolarization, right? Between depolarization and repolarization, right? This segment here, this green segment, that is what we define as being in the absolute refractory period. What that means is no new action potentials can be generated during that absolute refractory period. Right? Cannot trigger it. Right? So the absolute refractory period is a point in time when no new action potentials can be generated from within the axon hillock. Partly because you have not reached what? What has not been reached? between depolarization and repolarization, what has not occurred? Resting membrane potential. The membrane hasn't been reset. That doesn't happen until over here. Once you've reached hyperrepolarization, your possibility of going ahead and creating another action potential has happened. Why? Because you've surpassed resting membrane potential. You're beyond negative 70 millivolts. But between depolarization and repolarization, you haven't reached negative 70 millivolts. Action potential is not going to happen. Now, a relative refractory period will possibly allow for the excitation of a new action potential. And if you get a strong enough stimulus, in other words, boom, 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 you've overstimulated that area, then you can go ahead and you can actually drive another action potential. You can drive it. It'll happen. So within a relative refractory period, in other words, the period of hyperpolarization, as you return back to resting membrane potential, the potential of initiating an action potential does increase. I've seen it explained as if you think of refractory, if you think of absolute refractory as being a dog, right? And every time you call the dog is going to come back. If it's a well-trained dog, every time you call the dog's going to come back. Right? That's absolute refractory, except from the perspective of you can guarantee every time you try to initiate an action potential during absolute refractory, nothing is going to happen. Right? Just like with a well-trained dog, if you call the dog should come back. It's an absolute. Right? But within a relative refractory period, an action potential may or may not be generated. Right? With a not so well-trained dog, you might call the dog, and the dog might come back, the dog might not come back. Right? And so that's really the difference there. Right? So don't lose sight of that. Right? The next couple slides is just looking at differences in speed of the action potential between non-myelinated and myelinated axons. Again, this is something that I keep harping on, but it's an important difference that I want you to keep in mind. So, in a regular, in a just a normal bare bones plasma membrane, like what we see within the dendrites. Right? So dendrites are non-myelinated. Right? You have a stimulus. Right? You have the action potential, but as the action potential travels down the dendrite, it loses its potency. It loses its strength because dendrites are not myelinated. Right? So what happens? What's really happening during this period of time? Right? What's happening is sodium ions are leaking out. Sodium ions are leaking out. And as you lose sodium ions traveling down the dendrite, the strength of your action potential continues to decrease. Continues to decrease. And so in a non-myelinated axon, right, previous slide, this was a dendrite. Right, this was a dendrite. Right, so you can check that out right there. Right, but in a non-myelinated axon, what we see is we see repeated sodium channels all along the axon, even though there's no myelin. And so what happens? Right, the action potential is stimulated. Right, it starts to, because this is non-myelinated, so you start to lose electrical current. Right? But what happens? You hit another sodium channel. Boom. You hit another sodium channel. Boom. And so just as the strength of the action potential starts to wane, starts to decrease, what happens? You repropagate it and you build back up the strength. Right? And so that strength, while it starts to lessen, the frequency of the occurrence of those sodium channels are such that that you very easily repropagate it quickly. But here's what's happening in a myelinated axon. Right? So this is a myelinated axon. Right? You have your stimulus. Right? Notice that the strength of that action potential does not diminish very much at all, and it's moving over a greater distance than what it was in the non-myelinated. If I go back to this view, right, look how frequent those sodium channels are. Right? Here, remember, those sodium channels are only present within the nodes of Ranvier. That's it. Right? But this is able to go a further distance because the myelin sheath insulates. You're not losing the electrical conductivity. You're not losing the electrical charge to the environment because of the insulation. And then what happens? As soon as you're out of insulation, there is no ability to lose that charge because you're hitting more sodium channels. And so you're right back up to that full strength and you're moving it right on down again. And so you have fewer stops along the myelinated. You have fewer stops. And so you got a bunch of strong runners. Boom. All right, pass the baton. Boom. Pass the baton. Boom. Pass the baton. All right, as opposed to having a series of weaker runners who have to run shorter distances, and now you've got a delay of passing the baton. Run, delay of passing. So you're passing that baton more frequently, all right, than what you're doing over here. And that means that this message, this electrical message, stimulus, is going to reach its target, whether it's smooth muscle, skeletal muscle, cardiac muscle, or a gland. It's going to reach it a lot quicker. It's going to reach it a lot quicker. So just a couple more little things. I've actually only got like 2 more slides. I do want to talk a little bit about a type of neurons. All right. What we've really focused on so far are multipolar neurons. This is your typical peripheral nervous system neuron. Even central nervous system, these are relatively common. But that is not the only type of neuron that we have. And so here is your multipolar neuron. Let me this is an example of a multipolar neuron. All right, this guy right here, this is a bipolar neuron. All right, and then this one right here is a unipolar neuron. All right, what's the difference? Well, this is considered multipolar because here's your cell body, right, and each cluster of dendritic branching is a pole. So this is a pole, 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 and you've got a pole down here. So you've got multiple poles. Right? You've got multiple poles. With a bipolar, the cell body is directly in the middle, right? And you only have one dendritic branch and one axonic branch. One branch, two branch. One pole, two pole. Bipolar. Bipolar. Notice that the cell body is directly in the middle. It's directly in between the dendritic branch and the axonic branch. In a unipolar, the cell body is removed from the in between the axon. It's removed. It's on the peripheral. And so look, you've got your dendritic end. You've got an axon that is continuous with the axonic end. This is the receiving end. This is the processing end. But it's continuous. It's not disrupted by the cell body. And so therefore we look at this as being one continuous segment or one continuous pole. All right. Some of the things to keep in mind over here, look at the receptive end or the stimulus, the receiving the stimulus versus the moving of the stimulus, the conduction region, versus the secretory region or the response. In other words, the release of the neurotransmitter. Right? They differ for the multipolar, the bipolar, and the unipolar neurons. Right? They're different. Right? Here in the multipolar, right, the cell body with all of the dendritic branches, this is your receiving end. Right? This is what's receiving the stimulus. And then you've got your conducting area. This is where the action potential is going to occur. Right? And then you've got your secretory region down here. Right? This is where the neurotransmitters are going to be released. Right? Whereas in the bipolar region, right, this whole area from here all the way down to here, this is your trigger zone. All of this can be initiated by other dendrites of nearby neurons. Right? Remember, you've got differing types of synaptic connections that can occur between axons and dendrites. You have axodendritic, axosomatic, axoaxonic. And so any of these regions in here can very easily synapse with the axon terminals of nearby neurons. So all of this is the trigger zone. This then becomes the conducting zone. And again, only this region right down here will be engaged in the release of neurotransmitters. Right? And then the unipolar, right, it's very, very limited. Right? From here over, right, that's your receiving end. Right? This is what's going to be receiving the stimulus from nearby axon terminals. Right? But look, all of this is conducting. Right? And then this area down here, once again, this will be your area for secretion of your neurotransmitters. Right? And then this is just a nice little summary table, right, of your various types. And so I like to give you guys these just so that at least you have some further information in detail. So that was your crash course introduction into the overall organization and function of the nervous system. The next series of videos are actually going to be specific for the brain, right? The parts of the brain, function of the brain, and so we're actually going to look at that central nervous system a little bit more, and then you're gonna look at the autonomic nervous system, and then the embryonic development of the autonomic nervous system, which I think will be really interesting, and that'll be something that'll carry you right on through to AMP2, right straight through AMP2, all the way up to embryology and fetal development. So I think that'll definitely be a video for you to download and save. Let's say it like that. So with that said, go back, keep reviewing, keep checking your notes, keep practicing these questions, keep showing up, keep asking questions of me and of each other, and I'll catch you on the flip side.